Okay, in this video we're going to talk about techniques used to study gene expression in different organisms, specifically the technique of in situ hybridization, as well as a method called RT-PCR. So the thing is, it's often not enough to just know the genetic sequence of an organism. Instead, in a lot of cases, it's much more informative to know when and where a given gene is being expressed. And the same is true in other cases for knowing about expression of different clusters of genes. And so a method for tracking down where and when genes are being expressed is a technique that's called in situ hybridization. And the way this method works is basically you develop an RNA probe, so an RNA sequence of known sequence that is complementary to the RNA, messenger RNA produced when that gene is expressed. And when you develop these RNA probes, the probes are also developed and labeled with a fluorescent dye. So that relies very much on the same technique as I showed for the labeled nucleotides used in DNA sequencing earlier. And so then what you can do once you have a complementary RNA probe is then apply that probe to the organism that you're interested in and visualize where that probe winds up and kind of when that probe shows up and binds to something in the organism of interest. So I've got an example of an in situ hybridization technique applied to a fruit fly embryo. So in this case, the RNA that was prepared was complementary to a fly gene called the hunchback gene. And so when it was applied to these embryos, these are embryos at different ages. Hopefully what you can see is that the hunchback gene shows one expression pattern when the embryo is very, very young. And then as the embryo gets older, expression of that hunchback gene gets localized to particular regions in this fly and actually winds up having a very regular expression pattern as the fly gets older. So in situ hybridization is one great way to localize where a gene is getting expressed. This technique, again, can be extended to study the expression of groups of genes. If you have an RNA probe that is complementary to a bunch of those genes, but I'll note that to use this method, you have to know something about the organism's genome sequence first. What do you do if you don't know the organism's genome sequence? How can you get that information? Particularly if you can't directly sequence that organism. So another thing that was really discovered over the course of studying viruses was that viruses have this really interesting enzyme that's called reverse transcriptase. So if we think about what you know about the process of gene transcription, right, that's driven by RNA polymerase. Reverse transcriptase actually does something backwards. So rather than transcribing DNA, reverse transcriptase makes it possible to go from RNA back to DNA. So that's why it's called reverse transcriptase. It's the opposite of transcription. And this was discovered in a class of virus called a retrovirus. So there are actually a tremendous number of types of viruses that don't actually contain DNA. Instead, the viruses contain RNA. So kind of a classic example of a retrovirus is something like the HIV virus. So when retroviruses take over a host cell, one of the first things they have to do is they have to back convert their RNA to DNA. And so they contain an RNA sequence that codes for the production of this enzyme, reverse transcriptase. Then that reverse transcriptase back converts the virus's RNA to DNA, and then that virus can really take over the cellular machinery to make more copies of itself and spread itself further. And One really interesting thing about this is that if you use reverse transcriptase to go back from a messenger RNA sequence to a DNA sequence, when you do that, you're only going to wind up getting the coding sequence for that DNA, right? So you're not going to get introns or other untranslated regions, right? Because this is relying on the final processed messenger RNA product, not the original DNA sequence. And that can be incredibly useful because it tells you what actually gets expressed for a given gene. And you know you have an exact match with what's being expressed inside of a cell. So this also then can be coupled with the process of polymerase chain reaction to then amplify however many copies of that messenger RNA are present in a given cell. So basically, what you can do is, let's say you have a cell and you want to know, how many copies of a gene are getting expressed. The first thing you're going to do is you're going to back convert the messenger RNA that's present into cDNA with reverse transcriptase. And then you can isolate different messenger RNA sequences using the process of gel electrophoresis. And then you could potentially take those particular pieces of DNA and sequence them and figure out which genes are actually being expressed in a particular tissue at a particular age. So, that helps us figure out what genes are getting expressed, where and when, but that doesn't yet tell us too much about the function of genes. So we'll talk about that in the next video.